Oh,
Great.
nice.
Okay, so I'm online, and I can hear quite a bit of chatter, but it sounds like you guys can hear me okay too.
Thank you, Andy.
All right. Well, thanks everybody for your patience. Should we get going here?
Yes.
Great. Well, I want to welcome everybody to the first of what I hope will be a long series of C-DEBI networked speaker programs. Before I introduce today's speaker and we get started, I just wanted to say a few brief words about this program. There's a great challenge in having a distributed science and technology center in that our participants are scattered all around the US and all around the world. So in discussion among the executive committee and others organizing C-DEBI, we thought we'd like to try to use some of this new technology to bring people together across great distances. The other idea we really wanted to encourage was helping our some of our more junior colleagues to get exposure to the broad range of C-DEBI investigators and different people doing different kinds of work. And so we discussed ideally promoting the selection of postdocs or people that are relatively new out of graduate school in order to give them a chance to network and collaborate and also communicate to non-specialists. So if all goes well today, then we're hopeful we'll do another speaker in the winter quarter and we'll have another speaker in the spring. And I certainly encourage people listening in today to nominate themselves or nominate others to be named as C-DEBI network speakers. It's a bit of an honor to be selected for this program, and we'd love to get some additional young, energetic, and enthusiastic and brilliant scientists to tell us about their research. A little bit about logistics. There's a place for people who are participating to type in questions. I think we're going to have Brandi give her talk, and then at the end she'll take questions, but people are welcome to kind of put in their little chat comments as we go, and that way they can make sure they don't forget if there's particular issues that they want to raise. And then after the end of the presentation, Brandi can take some time to answer some of those questions. We're also trying to keep the talks to around 30 minutes. That way, hopefully, people will be more inclined to log in and participate. Finally, we're recording these, and they will be posted at the website so others can log in later and get to see what was said, get to see the visuals as well. So without any further delay, it's a great pleasure to introduce today's speaker, Dr. Brandi Reese, the University of Southern California. Brandi finished her PhD at Texas A&M earlier this year. She has a BS in geology, a master's in soil and water science, and then she started a PhD program in oceanography at Texas A&M. So she's just the kind of multidisciplinary research scientist that we think really brings a lot to the C-DEBI Science and Technology Center. She's interested in fields ranging from biogeochemistry to microbial, nutrient cycling, and biogeography. And her presentation today actually links these different topics together. So please welcome me in joining our first C-DEBI network speaker, Dr. Brandi Reese.
Thank you, Andy, for that introduction. And like you mentioned, I'll be taking questions at the end from the web audience. And thank you for this honor. And this is a work in progress. This is developing research. I'm hoping that I can get some good feedback about future directions, where to take this research. I'd like to start by thanking Heath Mills at Texas A&M University, as well as John Morse. Those are my advisors. All of this work was done in the Mills lab and partially done in the Morse lab as well. And of course, some other contributors, Scott Dowd, Alicia Shepard, Cruz St. Peter, Laura Zinke, for helping analyze and process these samples. And of course, Fumio Inagaki, Natasha Redinger, and Jonathan Gardner, who have all helped in sample collection. And then funding through IODP, NSF, and NOAA. We're all mostly familiar now with the new science plan for IODP going forward, starting in 2013. and the importance in the understanding of the microbiology, the geomicrobiology, and how it relates to the geochemistry. There are several major questions that we're trying to answer as a community of microbiologists, and more importantly on a global scale. So how active or how dormant are these communities? And how connected are these communities to the overlying water column? And I'd like to add to that, how connected are they to other locations in the, within the sediments and within the basalt for that matter? And last but certainly not least, but what function do these microbes have in the deep subsurface? So I'm hoping that you see today's talk as a hypothesis-driving tool and as a way to start communicating about how we answer these questions and how about the connectivity and about the pervasiveness of the microbial community. So with that said, what have we learned so far? Well, it's a widely known fact among C-DEBI community and among the greater IODP community that the subsurface biosphere accounts for up to 2/3 of the Earth's total biomass, or 66%. And that depends on what estimate you choose and what you look at. But it's also safe to say that in a majority of the places that we've looked for bacterial cells, we've found them in some form, even as deep as 1.6 kilometers in the subsurface. But also, depending on what study you look at, either bacteria are more dominant or archaea are more dominant. And the caveat with this is that with these reports and these studies is that they're using different methods to analyze this. So perhaps they're inadvertently selecting for or skewing their data in some way when they make their conclusions. Methods that we typically use to characterize the microbial life, they vary from lab to lab, and so we have all had these discussions about what's most cost-effective or time-effective or most efficient in terms of our output or our yield. But a shortcoming of most of these techniques is that you can't tell between live or dead cells, like microscopy or just inferring activity using radio tracers. Now there's methods like phospholipid profiling and cardfish and sequencing that can start to tell you the difference between living and dead. But at the end of the day, the only way you can tell who the in situ community is by sequencing. It's those A's, T's, C's, and G's that allow us to actually identify what microbe we're looking at. We've all heard over and over again about the Boss-Specking hypothesis. He stated that everything is everywhere, but the environment selects. Now, not trying to get into a philosophical discussion about this, but ultimately this is one of the questions that we're trying to address as C-DEBI scientists. We know that geographic isolation leads to communities of microbes that will evolve independently from each other. So their geochemical environment is impacting the microbial community, and likewise the microbial community is impacting their geochemical environment. So in answering this question, we ask how much these methods are being biased, our methods that we're using are biasing our results or are skewing our results. Are we looking at true biodiversity, or are we looking at different selection methods? So when we look at a biogeographic study, how much of it is just because we have chosen to look at a certain group inadvertently? Another question is, are we looking at the metabolically active community, or are we looking at the total population? And so it comes down to how we define the community. And it's important to look at the total population, and it's important to know the potential of the environment that you're looking at. And in doing so, that's why we use DNA targets. But if we want to know the metabolically active fraction in the population that's associated with changes in the geochemistry, then we should look at the RNA. And so for many reasons, including the difficulty of extracting RNA, and also the need to understand your total community first, DNA has actually been the target of most of these biogeographical studies. Within the past decade, this is just some previous studies that I went through and I looked at where they've characterized the subsurface microbial community, and they've used DNA targets. And a lot of it is by cloning the 16S ribosomal RNA gene. So this is an important distinction to make, that this is the ribosomal RNA gene and not the gene transcript, which is important to look at when you're trying to decipher these studies, is that the studies come up with different communities being the most numerically abundant or have the most common taxa in their study at these different locations. But an immediate question that popped up when I was looking at this was whether these communities are really different, or are the techniques that they're using, these different methods, altering their conclusions. Are we comparing apples to oranges, basically, by comparing total populations from environments that were not necessarily compatible? So we know that these were all sediment environments, but I asked, as a geochemist with a background in geochemistry, is what geochemical factors were affecting these communities. So remember, Boss-Becking said that the environment selects. But what that could actually mean is that the environment selects only the active population. So having to understand the geochemistry and assuming that it affects the community isn't a new study and has been known for a long time, but we can now better understand community comparisons by considering the fraction of the population that is affected and would be represented by the geochemistry. So that's where I take my focus, is looking at the RNA targets to really address what Boss-Becking was referring to. With similar processes available equal similar communities. So for those of you who are not familiar with RNA and DNA, let me give you a brief background to bring you up to speed. Just structurally, looking at it, DNA is double-stranded. RNA is single-stranded. So for this and then some other reasons as well, that this is a main reason that DNA is more stable and can be detected in cells that are dormant or are dead. It can also survive outside the cell for a matter of weeks or months, or even years in some cases. Whereas ribosomal RNA is degraded pretty quickly, and it's below detection limits outside the cells. And once a cell goes dormant or inactive, it will shut down everything but its most important functions in order to stay alive. And one of those functions is the transcription of RNA. So that's why dormancy leads to RNA that is below detection limits. So that's why we say that studies like this one that use ribosomal RNA gene transcripts are targeting the active microbial population. And that's the focus of the study and the work of the Mills lab at Texas. So to initially test this theory, I used multiple sites and I compared them for commonalities. Now this talk is a conglomerate of sites that have been undertaken over the course of the past couple of years. And the locations varied globally, but the thing I really want to point out and that I really want to stress is that the techniques and methods have stayed the same through all of these, throughout these sites and all these studies. So a sediment sample was, a half-gram sediment sample was extracted for ribosomal RNA, and then it is reverse transcribed back to complementary DNA, or C-DEBI. From there, it's PCR amplified using, and then visually looked at on the gel. It's always checked for contaminating DNA, and it's always checked for contaminants throughout the extraction process. And then the cDNA is sequenced using 454 power sequencing. So the beauty of this technology is that you can do multiple samples all at the same time using a tag or a molecular identifier. So a couple of things to note about this study is that all the samples were treated in the same manner and in relatively the same time in the same lab. Also, the B1 through B3 region was targeted for all of these samples, so that stayed consistent in using the 16S ribosomal RNA gene. And then also the PCR chemistry and the primers stayed consistent through all of this. And one more thing to point out is that all of this was done on the 454 PIRE sequencing in the same run. So, as I mentioned, the biases that tend to cloud many studies that focus on biogeography, where you can't tell if it is how much of it is real or how much of it is an inadvertent manufacturing just based on the methods. But in this situation, at least, all of the biases were carried through, so it's consistent throughout. The sequences were normalized for the number of sequences, and then they used the Ribosomal Database Project, or RDP, to align. I used Mother to train the sequences, and then cluster the OTUs, and all the annotation or the identification, your classification of your sequence was also done in RDP. And I used a Sorensen Diversity Index, which I'll show you here in a little bit, which is a beta diversity index to compare community similarities. And it also agreed with another diversity index called CARD. So using this approach and on those locations I mentioned, I was able to produce phylogenetic characterizations of the metabolically active subsurface populations. So first thing that I want to point out in this distribution, this is a phylum distribution in each location, is that each of these colors correspond to the same phyla. So the bright orange color shown here is Firmicutes. The pink color is Delta Proteobacteria. The purple color is Gamma Proteobacteria. But I'm not trying to get you caught up in all the individual groups, but rather just take a look at the big picture. These sites start to appear similar or different at the phyla level of classification. Now, I want to start by focusing on two sites that have relatively similar geochemical profiles with respect to oxygen, sulfate, methane, iron, and manganese. So theoretically, according to Boss-Becking, these profiles, since everything is everywhere, these profiles should look relatively similar. My geochemistry aside, though, I just want you to note, I do know the caveat that a lot of the chemical parameters are the same, but there are some that were different. So this is a loose characterization based on the geochemistry. So the Nankai Trough, first of all, this was leg 316 on the Chikyu. This is in collaboration with Tim Perlman, Natasha Redinger, and Rocky. Looking at the geochemical profile, what was interesting, well, first of all, we see sulfate that is reduced, and methane that is produced. So we have a sulfate-methane transition zone. Sediment was extracted above, within, and below the SMTZ. The first thing I noticed was that there was a greater diversity, number of OTUs, operational taxonomic units, at the surface and less at depth. At the surface, it was dominated by Delta Proteobacteria, and which you know is mostly sulfate reducers. And then it was dominated by Firmicutes towards the bottom, Beta Proteobacteria. Second study is the South Atlantic. This was done through a cruise through the University of Bremen and the Max Planck Institute on the RB Meteor. Again with Natasha Redinger and also Tim Perlman. These profiles were similar. You can see that there was also an SMTZ that was developed, and you see sulfates reduced. You can make out that there's methane that is produced at depth. And again, we have most abundant OTUs, least abundant towards the bottom, and we have a dominant firmicutes with some gamma proteobacteria. So based on these profiles are definitely distinct, but also they were very similar. But this is just at the phylum level. Now, a third study to kind of bring this into perspective is the Great Barrier Reef. This was Expedition 325. It was on the mission-specific platform, Great Ship Maya. And again, this is a bit of a contrasting site. This is a carbonate-dominated, it's a reef system. The sediments were more oxygenated. There was not an SMTZ. But we do see that there was a slight bit of sulfate reduction and a slight bit of ammonia increase towards depth. But we see that it's dominated by firmicutes, again, just looking at the phylum level. And so the key to comparing this comparison and the comparison between the other two samples is this level of resolution. Like I mentioned, we're at the phylum level. So realizing the potential for fine-scale differences, I did another analyses on these sequences. And that's when I applied some diversity indices and to give a comparison of the communities. So this is showing a pairwise comparison of each community. I've got a scale bar here of 10% community difference. There was a high resolution of sequencing allowed us to look at the biogeography to a greater detail. If you noticed, here we have these three sites, the Great Barrier Reef, South Atlantic, and Nankai Trough, which are all deep subsurface communities. They claded together. And here we have also, within one individual clade, are the SMTZ-dominated communities, the South Atlantic and the Nankai Trough. So another interesting find was, here we have the South Atlantic, the Nankai Trough, Great Barrier Reef, which grouped together. But then we also had the Gulf of Mexico hypoxic zone and the Palmyra Atoll. These are surface sediments. They were in the top 10 to 20 centimeters of sediment. They're slightly sulfidic, hypoxic to anoxic sediments. And those two began to group together. So if I take this from a global scale to a more local scale and look at just the Gulf of Mexico, I can look at biogeography on a local scale. This was a study, again, in the hypoxic zone, site AB5, 10B, and 8C, were sampled at noon and at midnight, and at a resolution of every 2 centimeters for a total of 20 centimeters. So all in all, there was a total of 58 samples that were collected. And this is just showing as an example of the phyla level with depth as a percentage of the total sequences for each of the sites. So this is representative, but all of the sites showed that no matter the east or the west, the phyla level, there really was no difference. Largely, the profiles all looked the same. They were homogeneous with depth, with time of sampling, with the seasonality, and location. But when I used a cluster analysis for the geochemical characteristics, I combined sulfate, chloride, sulfide, dissolved in reactive iron, by total organic carbon, total carbon, TRS, which is our pyrite, and grain size and porosity. I did this in SPSS and using JMP software statistical software tools. And I created a dendrogram using a neighbor joining algorithm. Again, 10% difference here on the scale bar. So I noticed first that each location grouped independently. Well, that makes sense. The location AB5, closer to the mouth of the river, was more productive, had slightly more hydrogen sulfide than the other two sites that were further from the mouth of the Mississippi River. Now, then I noticed also that there were clades based on depth, which again makes sense. We have changes in organic carbon with depth, with sulfide with depth, and the dissolved iron change with depth as well. But there was no statistical difference based on the time of sampling. So stayed on station, whether I sampled at noon or at midnight, there was no difference. Now, looking at the bacterial distribution here, I saw that there was, and I did again a pairwise comparison of the community, and which included over 264,000 genetic sequences in total. I saw that there was biogeography between the sites was most important, or was most apparent. And that was the primary clades that I grouped, which 10B, 8C, and 85. Well, that's good. It's showing that I did have some biogeography that was present. But then there was also secondary clade formation based on depth, the depth within the sediment. Well, that made sense, especially since we have slightly more organic carbon at depth and less surface. Okay. Well, then there was another clade development based on the time of sampling. So noon cores were distinctly different than the cores taken at midnight. And so even though it appeared that there was some diurnal variations in the microbial community, the geochemical structure and the statistics didn't support this finding. So because of this, I actually determined that I was seeing microheterogeneity in the cores at these sites. In other words, even though I collected cores at the same site 12 hours apart, I was never collecting the exact same community twice. So in that sense, on a much finer scale, I saw that there were differences at the molecular level and that there were actually community differences between one core. So what is the take-home message in all of this? Well, that biogeography using DNA confounds the issue molecularly. It makes it difficult to compare to the geochemistry. You don't know who is active and who is interacting with the geochemistry, or who is inactive. Now, again, remember I said that Voss-Becking said that the environment selects. Well, the environment selects at many levels, both locally and globally. And also, to better address the geochemistry with the molecular that we've chosen, an RNA approach. Now, I think it's important that we keep the methodology the same, but I also think it's important in order to be able to apply statistics and to look at things on a global scale for biodiversity, that we need to collect the same parameters between cruises. In order to compare apples to apples, we need to be able to collect the same geochemical parameters among each of the different research cruises. And so, I guess that leads to the questions that we have still to fill in to this bigger picture, which is the South Pacific Gyre and the North Pond, which have just been collected and so are heading back this way. And again, the South Pacific Gyre, which we have in hand and currently working on to be able to fit this into the bigger picture. So with that, if you have any questions, I'll be happy to take them. Do we have any questions from the web audience? And as Andy says, people should be, feel free to chat in your questions. So this, since this is for the C-DEBI network community, we're going to try to get the questions from the web audience first, and then we'll go to the audience here. Julie Huber. Were all these samples preserved in a particular way at sea, or were they dug out of the core repository? No, these were all preserved with the intent of molecular analysis at sea. So they were all preserved at -80. Well, I guess we can go ahead and take one more here in the audience before as these questions begin. Go ahead. We do have duplicates, and that is part of another ongoing is how much do duplication infect. Those are at the lab right now being sequenced as we speak. We should be getting that data back shortly. That was done as a separate study after these questions came up, and there was differences with how much that changes from among duplicates. All right, so we've got so we've got how deep the samples have come from. So the Palmyra and Gulf of Mexico were shallow, the top 10 to 20 meters. Great Barrier Reef, that was 40 meters maximum, so it was about 1, 20, and 40. For Nankai, those depths, at Nankai we've got 1, about 20, about 57. And with South Atlantic, it was right at the surface, 4, about 6.5, and about 8. For the day and night sampling, the genetic distance, genetic difference increased with distance. I believe that you're asking if. Honestly, I can't answer that question right now, but that is actually an interesting point. So with distance from each other, I'm guessing geographic distance is what you mean, genetic distance. Did the genetic differences change? And that's a good question. Yeah, geographic distance. Okay, so I'll have to go back and look at that, but that's an oh, that was Brandon Briggs. Myrna Jacobson Myers says, what are your plans for activity measurements? Well, each of these studies were not were independent of each other, and since they all came out of the same lab, that's when we started to look at how they interrelate to each other in a biogeographical sense. So the South Atlantic study, I know Natasha Redinger has done some activity measurements and has associated them to the South Atlantic in her study. With mine in the Gulf of Mexico, the work that I did do some sulfate reduction rates, compared them to the DSRA transcripts. I had some good agreement, and that's in a separate paper. Oh, okay. She's saying ribosomal RNA. You're talking about gene transcripts. Yes, and so yeah, that's what I meant for the Gulf of Mexico study. So that is definitely a future direction for these other studies. For the Gulf of Mexico study, I did do that. I have some transcripts for the DSRA gene, and as well I have that coupled to the rate measurements. I'm sorry, Jan? Yeah, there's, okay, yeah, with, Jan just asked what measurements are routinely done on on IODP cruises, and then what should be done. Well, what is attempted to be done and routinely done is doing measurements at your anions and your cations, and also organic carbon. There's and then as well as oxygen and some. I think that's covered most of the ones that are the major ones. There's some additional ones that individual labs will try to do on their own. And depending on the objective of the research or the objective of the cruise, the expedition, they'll, some of those will be scrapped and some additional ones will be added. So, for example, Pacific H Transect cruise, which is, they wanted to collect some microbiology on that cruise, but I couldn't really compare it because I didn't have the same measurements as I did, say, on mankind or. Rick Caldwell says, what is the current detection limit for extracting RNA from sediments? And are some sediments limiting in this regard? Yeah, there's definitely more sediments that are more difficult to extract. We noticed that the peat samples were more difficult. Those are more clay. They're a thicker sample, or just a more dense sample, so that we did have more difficulty particularly with clays. And the detection limit for extracting RNA. I've extracted RNA from, well, so far I have extracted RNA from South Pacific Gyre on a couple of samples that are in areas that are tended to fourth, tended to fifth, more on the fringe of the gyre. So I'm still working on the middle of the gyre, but had to test less. So just that's the best estimate I can give you for detection. We have a follow-up question from Yaron Dong. Has biomass or cell volume in the water core samples been measured? Yes, cell number. Has biomass or cell number in the water or core samples been measured? Yes, and we do have some of that data, and this does range from very dry sediments to and we do have for the cruises that are specifically for microbiology, I do have cell abundance. I don't have cell abundance for the cruises that were not in microbiology, like mankind. That was they wanted to preserve some for microbiology, but they didn't do those kind of measurements for. No, sorry, not for mankind, but for South Atlantic and for Pacific exploration. Some were preserved on. I do have those. Those numbers as well. Okay, we have one question from now. Asking how much time elapses between the time of collection, from the time it's in situ to the time it's preserved, to the time it is RNA is extracted, and then I'll get to your second one. But so when it comes on board, it's preserved almost immediately. I mean, there is some handling time, but it's within minutes. so it's very quickly, and it's at -8. These were not preserved with RNA later, straight -8. And then the extraction, these were extracted, so each of these varied just a little bit. Nankai was extracted within about 6 months. The South Atlantic is about a year. The Pacific H transect was in about a year. And then here for South Pacific Gyre, it's been 6 months. And so it varies, but we have gotten RNA out of samples that have been many years old, though, too. So, and your second part of your question was, which part? Or how much sediment do we need for the extraction? Well, our protocol calls for about 1/2 gram. So you can increase that a little bit up to about 1 gram, but 1/2 gram. Okay, and let's take one more question. We got it from Kiana Frank. Did your sequence co-registered 16S DNA-based community profile from the Gulf of Mexico sediment? How different was this profile? Was this from what you saw in your RNA-based community profiles? I did not do DNA. I have not done DNA work. All of my work is still in RNA. It would be interesting to look at how it differs with DNA. But not a question. And Jacobson Myers, okay, well, said we could talk about this later. She knows where to find me. I guess one more question from the audience, and then we'll wrap this up. Very good. Very, very deep. What do you expect in various layers? That's a very good question. And not to bias myself too much and to see what I'm expecting, but particularly heterotrophs in these environments. But yeah, it's a good question to I haven't seen any of the geochemical profiles for. So that's well, and looking at the active community. Can I repeat the very good question? Yes. I apologize about that, and everyone's going to learn and do better this next time. But Vika asked if what I expect from the South Pacific Gyre and the North Atlantic. These are going to be two toxic sediment columns, whereas the other two, South Atlantic and were definitely. I would like to, definitely we're going to be exploring that.